So when a doctor cuts into you, say you're having something removed from your body, or maybe you're having something like gallbladder surgery, the doctor will make an incision into you, and where he makes the incision, he may transfer some of the bacteria of your normal flora residing on the outside of your body to all of the organs and to your cardiovascular system when he uses his scalpel to cut into your body. So in that instance, you can have a potential chance of acquiring the opportunistic pathogen, which at one point wasn't pathogenic, but now that it's been moved to a new location, it doesn't really know how to react. And so a lot of times you end up getting a pathogenic type of response when you have something like that. Same thing, you don't even have to have surgery for that to happen. You could just get a simple cut, or you can, maybe have children and maybe they're skateboarding and they fall, or they're riding their bike and they fall and they cut themselves. So there are ways for bacteria to get in places where they're not supposed to be. And when they do that, especially for those non-pathogenic bacteria, they in a sense can become pathogenic because they can induce infection that way. And so, but for the most part, most pathogens that are considered to be opportunistic pathogens, they're not pathogenic and they don't cause disease. They only cause disease when given the opportunity, which is why we call them opportunistic pathogens. Now, pathogens can bring on two different types of diseases, or two groups of diseases. At least this is how we classify them. We have infectious diseases, and then there's microbial intoxications. Infectious diseases occur when you have a pathogen, a bacteria, a fungus, or a protus that colonizes a person's body, and then once it colonizes, it's going to start infecting tissue and inducing disease inside of that host. And so this is what we refer to infectious disease because it infects the tissue of the body. Now, when we talk about microbial intoxication, we talk about primarily when you have a pathogen that resides outside of the host it's going to infect, and in some way that pathogen is able to get some of its toxic components into the host, which causes the host to become very sick. One example we have here would be that of Clostridium botulinum, which is an organism that can cause individuals to develop botulism and make you very, very sick. So you have a bacteria, the Clostridium botulinum, that can live in environments where there's a high concentration of proteins in something like meat, and that meat hasn't been canned properly, or it hasn't been processed properly. So it develops very good conditions for the Clostridium botulinum to seed itself. And when that happens, the Clostridium botulinum starts producing what we call the bot toxin. And the bot toxin can lead to botulism once it's been ingested. So essentially what we have here is a pathogen that has been produced in a sense in vitro, or it's been produced outside of the body in some environmental structure. So in this instance it would be that of a can, and when it's living in this particular environment, it will start to produce a toxin. A person that goes to eat some of the infected meat is going to ingest the toxin, and then that toxin will elicit the disease inside of the human body. Okay, so this is what we mean when we talk about microbial intoxication. You have components taken from a bacteria or produced by a bacteria that somehow make their way into a host system. It could be human, it could be different types of animals, but somehow in some way that toxin ends up getting inside of the host, and then disease is elicited via the toxin. So with microbial intoxication, you have disease being elicited elicited by a toxin. With infectious disease, you have disease being elicited by the actual pathogen itself. So let's kind of review now, the reasons why, other than the fact that this is your last step that you need to take to get into the dental hygiene program. Let's talk about some of the reasons why we want you to, study microbiology for the sake of, this course, and hopefully for the sake of, somewhere down the line, hopefully you'll find some of the information you've used in this class very useful. But we study microbiology because we can't get away from microbes. They're everywhere. They're ubiquitous, which means that they're all over. And they play a very significant role in our lives. They're essential for the life on this planet. A lot of the oxygen that was first generated on Earth stemmed from microorganisms that contain small chloroplasts, and they released oxygen into the environment, which gave rise to a lot more complex organisms than what existed at the time. And so those types of organisms were actually photosynthetic algae, as well as some photosynthetic bacteria. The primary one was referred to as cyanobacteria. And they were able to give rise and really to flourish because there were less living entities at the time, in the early days of the planet. Because of that, you just had this abundance of oxygen, and the abundance of oxygen eventually gave rise to more complex organisms. Microorganisms also assist in the cleanup of our environments. They are very useful in the decomposition of dead organisms as well as waste products. So without microorganisms, we'd have a high concentration and a large buildup of a lot of dead material as well as waste products that are out there. So we can use microorganisms in a form that we refer to as bioremediation, where we use them, we grow them in a great abundance, and then we apply them to some of the waste that we as humans have generated in this planet. And we're trying to use the microorganisms to degrade that waste so that way we can get rid of it. Now, there's a specific term that we use to refer to organisms that thrive on dead organisms or are able to live off them. The primary example would be that of fungi, but the term we use to refer to them as is saprophyte. And saprophyte just means that this is a type of organism that is able to live and digest organic material. And as I said, we use microbes to clean up toxic waste, and this is referred to as bioremediation. So here's an image outlining a microbe as a saprophyte. I told you the primary ones really are fungi. You can see these if you go on hikes, or if you're just hanging out in the woods, or if you kind of walk around York Tech right now, you might be seeing some of the fungus that's out there. Fungi are pretty cool. We're not going to spend too much time talking about them, but they are among the key examples of what a saprophyte is. What they tend to do is they will affix themselves to decaying organic material. Tissue of this wood, tissue of these leaves, tissue of a human's foot. What happens is the fungus basically can penetrate itself into the tissue, and when it does that, it releases a bunch of digestive enzymes into the tissue, be it this hard wood here, be it these leaves, be it a human patient's foot, and the release of those enzymes help to break down that material. So in conjunction with extracting resources from the decaying organic material, these fungi can also extract resources or nutrients from the soil or substrate that they're affixed to. So they can also absorb nitrates, phosphates, sulfates, and then they will also produce carbon dioxide, because these guys, unlike bacterial cells, are actually eukaryotic. And being eukaryotic, they're heterotrophs. primarily, which means since they're feeding off something, they're going to release waste, and they're going to release waste in the form of carbon dioxide. So microorganisms have some very specific roles. They help assist in elemental cycles, such as the carbon cycle, the nitrogen cycle, oxygen, sulfur, phosphorus cycles. They play a huge role in making sure that these chemical elements basically help to circulate and enable life to thrive, because we can't get away from these chemical elements. We need them to survive as well, as do a lot of other living organisms. So microorganisms play a very great role in ensuring the presence of these specific, the presence as well as the utilization of these specific chemicals. Some other roles of microorganisms: you have algae and bacteria. They help to serve as a diet source for many animals, so they play a very strong role in the food chain. Algae, bacteria, all of those algae. We eat algae all the time. If you go to Whole Foods or Mom's or Earth Fare, any of those places, you can find a lot of algae that we've tried to convert to so that our bodies can absorb nutrients, so that way we can live longer, because they're finding ties to eating healthier through the consumption of different types of algae. One in particular is called spirulina. If you've never eaten that before, it's supposed to help kind of as a dietary supplement, as well as to provide you with a lot of vitamin A. So there's different types of algae out there that play a role in assisting very small animals to survive, but we also as humans are looking to these types of this particular group to extract vitamins from the organism. Bacteria, we use bacteria in yogurt primarily. That's probably one of the best things that we use bacteria for. We also use fungi to generate wine and different alcohols. So in a sense, they serve as our food source, as well as food source for other living organisms as well. Microbes that reside in intestinal tracts of animals also play a very significant role. They help assist in the digestion of food. Your entire intestine is lined with a lot of E. coli, and that E. coli helps you to break down any food that passes through that system. If you didn't have your bacteria, it would take us a lot longer to break down food components and enable us to turn that food into energy for our cells. And so they're very beneficial in assisting with the processing of food, so we can extract nutrients from the food that we eat. Microorganisms are also very useful in the research setting. We like to observe them as a cell model, which means their cells are less complex than our own. And because they are, we use them to hopefully understand more about our own cells. Take a bacterial cell, for example. There's not much in a bacterial cell. You have standard structures such as cell membrane, you have cytoplasm, you have ribosomes and proteins, you have a nucleoid region, which is essentially just a circular DNA. And then you also have, and then you also have cytoplasm, if I didn't say that already. So you don't have very much inside of a microorganism or a bacterial cell, but you have a lot more structures in a eukaryotic cell, which is what our cells are made of, or the type of cell we have. So the more we know about microbial cells, the more we start to comprehend about our own cells and start to kind of put together where it is we think that we may have originated from, or where some of the earlier cells came from. So I just want to go back to this idea that this first bullet point here of microbes playing central roles in various elemental cycles. To give you an example, we have that of microbes serving as a